I want you to do something for me right now. Just take a deep breath in and hold it for a second.
Let's hold it right there.
Yeah. While you're holding that air in your lungs, I want you to really think about what is actually happening inside your chest.
There is a lot going on.
Right. Like what invisible automatic processes are just firing off in the background, making sure you don't literally suffocate.
It is a lot to think about.
Okay, you can exhale now. Go ahead.
Yeah, please breathe. But, it's an entirely seamless process from our perspective. We rarely give it a second thought.
Almost never.
Right. But behind that simple feeling of taking a breath is this incredibly sophisticated feedback loop.
It's wild.
It really is. It is this precarious split-second balancing act between these massive mechanical pumps and incredibly delicate blood chemistry.
And that balancing act is exactly what we are decoding today. Welcome to our custom deep dive into a really fascinating stack of sources you brought in.
I am excited for this one.
Same here. We're looking at a combination of advanced respiratory physiology notes, some clinical acid-base interpretation guides, and some absolutely stunning histology slides.
Oh, the slides of the intrapulmonary air conduits are just beautiful.
They really are. And the mission here is to figure out how this mechanical and chemical cascade keeps you alive, and frankly, how doctors play detective to untangle this whole system when it breaks down.
Absolutely. And to really understand how clinicians interpret the complex chemistry of arterial blood,
Yeah.
we can't just jump straight into the math.
Right, you need the context.
Exactly. We have to start with the physical machinery that brings those gases into the body in the first place. We have to look at the pump.
Okay, let's unpack this. If we look at the physical mechanics of getting air into the lungs, it's essentially a set of organic bellows.
That is a great analogy.
And the diaphragm is doing the heavy lifting here. The physiology notes point out that during quiet breathing, the diaphragm is doing like 75% of the work of moving gas.
The vast majority of it.
But what blew my mind is the economy of movement. I mean, it only drops about 1.5 centimeters to make a breath happen.
Yeah, that 1.5 centimeter drop is deceptively powerful.
It doesn't sound like enough to do anything.
Right. But when the diaphragm contracts and flattens downward, it increases the vertical dimension of your thoracic cavity.
Okay.
So by expanding that volume, the pressure inside your lungs drops below atmospheric pressure. It is just Boyle's law applied to human anatomy.
So pulling the bellows open drops the pressure.
Exactly. The outside air is at a higher pressure, so it naturally just rushes down the pressure gradient directly into your lungs.
Oh, okay.
And the diaphragm does have a little help on the inhale. The external intercostals contract to elevate the ribs, which expands the chest anteriorly.
Right, so the inhale is this active, energy-consuming process, but the exhale is a totally different story.
What's fascinating here is the sheer mechanical efficiency of the human design.
How so?
Well, during normal quiet breathing, expiration requires zero active muscle contraction. Zero. The diaphragm simply stops receiving the signal to contract, and it relaxes.
And then the lungs just, what, snap back?
Pretty much. If you look at those histology slides we mentioned, you see the lung parenchyma is loaded with elastin fiber.
Okay, yeah.
So the elastic recoil of that highly strecky lung tissue just pulls everything back to its resting state, passively pushing the air back out.
Wow.
It's completely free energy.
I love the idea of free energy in biology, but obviously that passive system only works when we are, just sitting around doing nothing.
Right, resting state.
Because when you switch to deep or forced breathing, like if you're in the middle of a heavy deadlift or if a patient is battling a severe respiratory infection, those passive mechanics go out the window.
They absolutely do.
The sources talk about accessory muscles coming online. To forcefully inhale, you've got the scalenes in your neck pulling up the first two ribs.
Yeah, and the sternocleidomastoids hauling the sternum upward.
Exactly. And suddenly, expiration isn't free anymore either, right?
Not at all. To actively force air out, your abdominal muscles and your internal intercostals have to aggressively contract to squeeze that thoracic cavity.
Which has to burn a ton of energy.
Oh, it requires a tremendous amount of ATP. It's a massive metabolic cost just to push air out of the body when you are stressed.
But having this incredibly strong physical pump is completely useless if there's nothing driving the rhythm.
Right, a pump needs a motor.
Exactly. The muscles need a maestro. They need a signal telling them exactly when to fire and how hard. And that control room isn't anywhere near the chest.
No, it's sitting up in the brainstem.
Right.
It's the neurological command center. Specifically, we're talking about the medulla oblongata and the pons.
And the organization here is brilliant, according to the notes.
It really is. Down in the medullary center, You have two highly specialized clusters of neurons. The dorsal respiratory group, or DRG, acts as the primary pacemaker for inspiration.
Yep.
It sends the electrical signal down the phrenic nerve to tell the diaphragm to contract.
And then there's the ventral respiratory group, the VRG.
Yes.
But the sources note that the VRG handles expiration, which I have to admit kind of confused me at first.
How come?
Well, if expiration is usually passive and just relies on elastic recoil, why does the brain need a dedicated neural cluster for it?
That is a great question. It acts as an overdrive gear.
In an overdrive gear. Okay.
Yeah. During quiet breathing, the VRG is essentially dormant. It's not doing anything.
Because the recoil is doing the work.
Exactly. But the moment you start exercising, or if a disease process increases your airway resistance, the passive recoil just isn't enough.
So the VRG kicks in.
Right.
Yeah.
The VRG wakes up and starts firing signals to those accessory abdominal muscles we just talked about, forcing the active exhale.
Okay, so the medulla basically has our baseline gas pedal for inhaling and this overdrive gear for exhaling.
Correct.
But it's taking orders from the pons, which sits just above it in the brainstem. And the pons seems to be like the fine-tuning mechanism.
It provides all the nuance. The pontine center has two distinct areas. The apneustic center promotes deep, prolonged, gasping inspiration.
Okay.
It essentially constantly stimulates the DRG to keep inhaling.
Which sounds incredibly dangerous if left unchecked, like you just inflate like a balloon.
You would, which is exactly why the pons also houses the pneumataxic center.
And that's the limiter.
Yes. It overrides the abducystic center, controlling the rate and physically terminating the inspiratory signal.
Here's where it gets really interesting. The brain has this internal tug of war regulating the breath, right?
Yeah.
But it also has an external failsafe wired directly into the lungs themselves. The sources detail the Herring-Brewer reflex.
Yes, the ultimate biological brake pedal.
Right. The lungs are embedded with stretch mechanoreceptors.
Yes, they are.
When the lungs inflate to a certain physical threshold, these receptors fire a signal up to the brain that basically says, stop inhaling immediately.
They cut it off.
But I'm trying to wrap my head around why we evolved a physical kill switch for breathing. What are we protecting against here?
Barotrauma.
Yeah.
Pure and simple.
Oh, wow.
Think back to those microscopic cross-sections of the intrapulmonary air conduits in your histology slides.
The really thin ones.
Exactly. The terminal bronchioles and alveoli are phenomenally delicate single-cell thick structures.
Right.
If the apneustic center malfunctioned and the diaphragm just kept pulling downward, the sheer negative pressure would quite literally rip those microscopic tissues apart.
That is terrifying.
It is.
Yeah.
So the Herring-Brewer reflex protects the physical integrity of the lung architecture. It's especially vital in infants to terminate normal inspiration, and it kicks into high gear for adults during intense exercise or when dealing with hyperinflation diseases like COPD.
Okay, so the brain controls the rate, and the stretch receptors protect the hardware.
Yep.
But how does the brainstem actually know when to speed up the breathing rate or slow it down? It's not just running on a static timer, obviously.
Well, not at all.
It's constantly adjusting based on chemical sensors that are sampling the blood.
The respiratory system is fundamentally a chemical management system, and the body uses two distinct sets of sensors, central chemoreceptors and peripheral chemoreceptors.
Okay, let's start with the central ones.
The central chemoreceptors are sitting right there in the medulla. But here's the trick. They are bathed in cerebrospinal fluid, not blood.
Which the sources highlight as a crucial distinction.
It is. CSF has very little protein buffering capacity compared to blood.
Wait, let me make sure I understand the implication there. Because the fluid around the brain doesn't have chemical buffers.
Right.
Any slight change in carbon dioxide levels causes an immediate massive swing in the fluid's pH.
Precisely. The central chemoreceptors are exquisitely sensitive to an increase in the partial pressure of carbon dioxide, or PCO2, and the resulting drop in pH.
Okay.
They detect that acidity instantly and tell the lungs to hyperventilate to blow off the excess CO2.
And while the brain is monitoring its own fluid, the peripheral sensors are out in the body's main plumbing.
Oh, the arteries, yeah.
You've got the carotid bodies located in the carotid arteries, sending data back via the glossopharyngeal nerve. They monitor oxygen, carbon dioxide, and pH.
Correct.
And then you have the aortic bodies on the aortic arch, reporting via the vagus nerve. But the sources highlight a really strange quirk here.
The pH blind spot.
Yeah. The aortic bodies monitor oxygen and carbon dioxide, but they completely ignore pH. Why the division of labor? Like, why would the aorta not care about acidity?
It all comes down to prioritizing the brain. The carotid arteries are the direct supply lines up to the brain.
Oh, right.
The carotid bodies must monitor pH because an acidic shift can severely disrupt neurological function.
That makes total sense.
But the aortic arch, on the other hand, is monitoring the systemic mixing of blood heading out to the rest of the body.
So it had a different job.
Exactly. It cares about the raw gas delivery, oxygen and CO2, but it leaves the strict pH regulation to the carotids and the brain itself.
That is brilliant. But looking at all these sensors together, the clinical notes emphasize a point that feels backwards.
How about the oxygen?
Yes. Under normal conditions, a lack of oxygen is actually not what makes you feel like you need to breathe.
No, it isn't.
The absolute strongest drive to take a breath is a buildup of carbon dioxide.
Rising PCO2 is the primary alarm bell. But there is a massive clinical exception to this rule, and it completely rewires how doctors have to treat certain patients.
You're talking about the COPD switch, the hypoxic drive.
Yes, patients with severe chronic obstructive pulmonary disease have chronic hypercapnia. They constantly retain carbon dioxide.
Because they can't exhale it properly.
Exactly. So over years, their central chemoreceptors are bathed in so much acidic fluid that the receptors actually become desensitized.
Wow.
The brain essentially gets used to the CO2 alarm ringing all day, and eventually it just unplugs the alarm entirely.
So if the brain ignores high carbon dioxide, how does a COPD patient even know when to breathe?
The primary drive to breathe physically shifts. Their brain starts relying entirely on the peripheral chemoreceptors, sensing low oxygen levels.
Just the low oxygen.
Right. That becomes their only trigger to take a breath.
Which sets up a terrifying scenario in an emergency room.
Oh, absolutely.
If a COPD patient comes in struggling to breathe, the natural instinct of a paramedic or a doctor is to slap a mask on them and give them 100% pure oxygen.
Of course it is.
But if their brain relies solely on low oxygen to know when to breathe, does giving them pure oxygen literally tell their brain to stop breathing?
This raises an important question, and it is one of the most classic high-stakes dilemmas in emergency medicine.
I can imagine.
If you flood a severe COPD patient with highly concentrated oxygen, you can completely knock out their hypoxic drive.
That is wild.
The peripheral chemoreceptors sense all that abundant oxygen, signal the brainstem that the emergency is over, and the patient's respiratory effort can drop dangerously low.
And then the CO2 builds up even more.
Exactly, causing their carbon dioxide levels to spike to lethal levels. It's why supplemental oxygen for these patients has to be titrated with incredible precision.
You have to thread the needle.
Right. You want to give them just enough oxygen to survive, but not enough to turn off their drive to breathe entirely.
That is crazy. You can over-satisfy the biological sensors to the point of danger.
You really can.
And you can manipulate the system in the other direction, too. The sources note that conscious hyperventilation blows off massive amounts of carbon dioxide.
Right, which drops the CO2 levels.
And by stripping the CO2 out of the blood, you essentially silence the central chemoreceptors. It's the exact mechanism freedivers use to hold their breath for 5, 6, 7 minutes underwater.
Yes.
They trick their medullary sensors into thinking everything is perfectly fine because the CO2 hasn't built back up yet.
They are temporarily hacking the feedback loop. But the reason those chemoreceptors are so paranoid about carbon dioxide and pH in the first place is that human cellular function is incredibly fragile.
How fragile are we talking?
Very. Enzymes denature, ion channels fail, and cardiac muscle loses its contractility if the blood's pH strays far from 7.4.
So the margins are tiny.
Tiny. Which brings us to the clinical core of your source material, which is acid-base interpretation.
Right. Decoding the chemical wreckage when the system fails. The sources lay out 4 primary disorders doctors are hunting for.
Respiratory acidosis, respiratory alkalosis,
and metabolic acidosis and metabolic alkalosis. And to figure out which one is wreaking havoc, clinicians use a strict 3-step checklist.
Step 1 is establish the primary disturbance by looking at the pH, acidemia, or alkalemia.
Step 2, if it's metabolic acidosis, determine the serum anion gap. And step 3, evaluate the compensation.
I think the concept of compensation is where the true elegance of the human body really shines.
Okay, let's use an analogy here to help visualize it. I picture this entire system as a chemical seesaw.
Okay, I like that.
On one side, you have the metabolic system, the kidneys and tissues dealing with bicarbonate, which is your base.
Right.
On the other side, you have the respiratory system, the lungs dealing with carbon dioxide, which acts as your acid.
Perfect.
If your metabolism starts failing and dumps lactic acid into your blood, your pH drops. The metabolic side of the seesaw hits the floor.
It's a great visualization. But what makes the seesaw complex is the element of time.
Right, the speeds.
Exactly. The two sides do not move at the same speed.
Because if the metabolic side hits the floor, the lungs immediately kick in. Like within minutes, the respiratory system starts hyperventilating to blow off carbon dioxide.
Yes.
They are literally exhaling the acid to lighten their side of the seesaw and pull the pH back up toward normal.
Exactly. The respiratory compensation is very rapid.
Right.
But if the primary problem starts on the respiratory side,
Say severe asthma or COPD.
right, causing carbon dioxide retention and respiratory acidosis, in that case, the metabolic side has to respond. The kidneys will recognize the acidic shift and begin to enact a renal compensatory mechanism.
What does that look like?
They reabsorb and generate new bicarbonate to offset the acid. But unlike the lungs, the kidneys are very slow.
How slow?
This renal compensation takes 3 to 5 days to fully engage.
So time is a massive factor. But the doctors can't just guess if the seesaw is balanced. They have to do the math to prove it.
They absolutely do.
And that brings us to Winter's formula. The sources highlight this as a critical tool when a patient has metabolic acidosis to check if the lungs are actually hyperventilating exactly as much as they should be.
Yes, the formula is expected PCO2 equals 1.5 times the bicarbonate level plus 8 plus or minus 2.
It is an absolutely essential calculation for a clinician.
It really is. Let's walk through why. Imagine a patient comes in with a severe metabolic acidosis. Their bicarbonate is extremely low. Okay. You run Winter's formula to see where their carbon dioxide should be if their lungs are compensating properly. But when you look at their actual lab results, their carbon dioxide level is significantly higher than the expected range Winter's formula gave you.
So they aren't blowing off enough CO2.
Exactly. And that tells the doctor something vital. The patient doesn't just have one problem. They have a concomitant or simultaneous respiratory acidosis sitting right on top of their metabolic acidosis. Their lungs are failing to compensate.
Which points to a really crucial rule from the clinical guide, one that seems counterintuitive at first. Compensatory changes never entirely correct the primary disorder back to a perfectly normal pH of 7.4.
Never. The body's defense mechanisms are designed to mitigate the immediate life-threatening danger, but they don't erase the primary footprint of the disease.
So if a doctor looks at a lab report and sees a beautifully normal pH of 7.4, but totally deranged carbon dioxide and bicarbonate levels, that isn't a sign of a healthy, fully compensated patient.
No, that is a massive red flag.
It suggests a mixed disorder, doesn't it?
Yes, it means two independent primary problems are happening simultaneously, artificially canceling out each other's effect on the pH.
The sources use the brilliant example of salicylate intoxication for this, a massive aspirin overdose.
It is the classic example.
What exactly does a massive dose of aspirin do to the seesaw?
It attacks both sides at once. Salicylates directly cross the blood-brain barrier and heavily stimulate the medullary respiratory center.
So it forces the patient to hyperventilate?
Yes, which causes a primary respiratory alkalosis. But simultaneously, the toxic aspirin levels uncouple oxidative phosphorylation in the cells.
Meaning?
It forces the body to produce massive amounts of organic acids, causing a primary metabolic acidosis.
So you have a primary respiratory alkalosis pulling the pH up, and a primary metabolic acidosis pulling the pH down.
Exactly.
The final pH might look confusingly normal, but the underlying chemistry is a complete disaster.
Precisely. If you don't meticulously follow that three-step interpretation process and run the formulas, you will miss the toxicity entirely.
So what does this all mean for the poor resident doctor tasked with gathering this data? To run Winter's formula or uncover a mixed disorder, they need incredibly precise numbers.
They really do.
And you can't get that from a standard venous blood draw on the arm. Veins carry blood that has already passed through the tissues. It's like the exhaust pipe.
Right.
Doctors need to see the raw, unadulterated delivery data just as it leaves the heart. They have to go into the arteries.
Yes, the arterial blood gas, or ABG. It's a highly specific and quite uncomfortable procedure.
I've heard it hurts.
It does. The blood is typically drawn from the radial artery, right at the wrist where you'd feel a pulse. But before a needle ever touches the skin, the clinician must perform the modified Allen test.
Because if you damage the radial artery, you could theoretically compromise blood flow to the entire hand.
Exactly. So the clinician physically compresses both the radial and ulnar arteries at the wrist, temporarily stopping blood flow to the hand.
Okay.
Then they release only the ulnar artery. They watch to see if the hand flushes pink again.
Ah.
They have to physically prove that if the radial artery spasms or forms a clot after the needle puncture, the ulnar artery is robust enough to keep the hand perfectly perfused on its own.
It really is a high-stakes draw. Once the Allen test is clear, the collection uses a standard 3 milliliter syringe, but with a very fine 25-gauge needle, and the syringe preparation is totally unique. The clinician aspirates 1 to 2 milliliters of liquid heparin, an anticoagulant, into the syringe, and then pushes it all back out.
Leaving the plunger fully depressed.
Right.
That microscopic coating of heparin left on the walls of the syringe is critical. It prevents the blood sample from microclotting before the lab machine can analyze it.
And the actual draw is entirely passive, isn't it?
Yes, because arterial pressure is so high, the moment that needle finds the center of the artery, the pulsing blood pressure will automatically push the plunger back and fill the syringe on its own.
The sources specifically call out the use of that tiny 25-gauge needle. It's not just for patient comfort. It's to prevent traumatic hemolysis.
Yes. Red blood cells are essentially bags of potassium.
Okay.
If you force blood aggressively through a needle or a catheter, the sheer physical force can rupture the cell membranes.
And if they burst?
If those red blood cells burst, which is hemolysis, they spill all their intracellular potassium directly into the plasma sample.
Which skews the data.
Oh, completely. The lab will report a terrifyingly high potassium level, but it's an artifact. The patient isn't hyperkalemic, the sample was just handled too violently.
Wow.
You need pristine, unruptured data to do the math.
And once you have that pristine data, the real mathematical heavy lifting begins. Step 2 of that checklist we mentioned earlier, the serum anion gap.
Yes.
The calculation is straightforward. Sodium minus the sum of chloride and bicarbonate. Historically, a normal gap was considered to be 12 plus or minus 2.
Right.
But the clinical notes highlight a fascinating shift in modern medicine that actually changed that breast line.
It's a perfect example of how underlying technology dictates clinical parameters. The older lab machines used colorimetric assays to measure serum chloride.
Okay.
But modern labs use highly sensitive, ion-selective electrodes.
So the modern machines are actually capturing more of the chloride that was already there.
Exactly. Because the modern machines measure a higher concentration of chloride, the number you are subtracting in the anion gap equation is larger.
Ah, I see.
Therefore, the normal range for the gap on modern autoanalyzers has mathematically shrunk to between 3 and 9 milliequivalents per liter.
That is a huge difference.
It is. If you use the old baseline of 12 on a modern machine, you will completely miss a subtle metabolic acidosis.
If we connect this to the bigger picture, the anion gap isn't just an arbitrary math problem. It is a diagnostic spotlight.
It absolutely is.
If the gap is elevated above that 3 to 9 range, it definitively proves the existence of unmeasured organic acids circulating in the blood.
Things like lactic acid from sepsis, ketones from diabetic ketoacidosis, or toxins from an overdose.
It gives the physician a concrete starting point to find the root cause of the chemical collapse.
Right.
And they don't stop there. Clinicians use other advanced formulas to complete the puzzle. The osmolol gap to hunt for toxic alcohols, the delta-delta calculation to definitively prove the existence of triple mix disorders,
And the alveolar gas equation to determine if the lungs are failing to transfer oxygen properly. Interpreting these values requires a masterful synthesis of physiology and pure mathematics.
It really does. We have covered an immense amount of ground today.
We really have.
We started by looking at the pure mechanical bellows of the chest, the diaphragm dropping that crucial 1.5 centimeters, and the passive free energy recoil of expiration.
Machinery.
Right. We moved up into the brain's control room, looking at how the medulla and the pons coordinate the rhythm while the lungs' own stretch receptors protect the microscopic tissues from barotrauma.
And the sensors.
Yep, we zoomed in on the central and peripheral chemoreceptors, exploring the clinical danger of the COPD hypoxic drive.
The oxygen paradox.
And finally, we saw how doctors extract a heparinized syringe of pulsing arterial blood to use equations like Winter's formula to decode the exact state of your chemical seesaw.
And the core realization to take away from all these sources is the profound interconnectedness of human biology.
It's all linked.
It is. The physical architecture of the lungs, the neurological wiring of the brainstem, and the metabolic function of the kidneys are not isolated systems. They are locked in a constant vital chemical dialogue, shifting and compensating second by second to maintain the fragile conditions required for life.
Which leaves you with a really provocative thought to ponder. We just discussed how conscious hyperventilation can temporarily silence our medullary chemoreceptors by blowing off carbon dioxide.
Right, manipulating the feedback loop in real time.
But it makes you wonder if our breathing mechanics can alter our blood's pH so dramatically in the short term. Could specific, disciplined, long-term breathing practices permanently hack our metabolic acid-base baseline?
That is a fascinating question.
Could we consciously train our own chemical seesaw to operate with greater resilience? Something to think about the next time you take a deep breath.